I'm Adam Rivers, a computational biologist for the USDA's Agricultural Research Service. In this tutorial, I'll be explaining how to use amplicon sequencing and the software package QIIME2 to analyze fungal ITS sequences. The amplicon sequencing is a very cost-effective way to profile the composition of a microbial community. In fungi, the commonly used gene region to do this in is the internally transcribed spacer. This is a region between the small subunit ribosomal RNA and the large subunit ribosomal RNA. In between these two genes is the 5.8S ribosomal RNA gene. So the ITS2 sequence is broken up into the ITS1 region, which is between the small subunit ribosomal RNA and the 5.8S, and the ITS2 region, which is between the 5.8S and the large subunit. Now, the size of these two regions is about 180 base pairs, and the total ITS region is about 650 base pairs, including the 5.8S. But there's a lot of variability in the length of these regions. As you can see from the plot above, there are these ITS regions can go as far as, be as large as 2,500 base pairs, and so there's a real variability. There's also a wide range of sequencing primers that are used to analyze ITS, to amplify ITS sequences. And this wide range of primers share one thing in common. They're all anchored in these conserved regions. So in contrast to the 16S region, which is commonly used for fungi and for bacteria and archaea, the ITS has several things that are different. The length is very variable, and it has these highly conserved regions and these highly evolutionarily divergent regions. And so this variation causes some challenges. And that means that we need to oftentimes trim the ends of the regions and then think about how we're identifying the actual sequence variants in the sample. So for this tutorial, what I've used is two samples that come from an experiment that was done in May's Microbiome Science. And this experiment actually looks at the effect of winter cover crops on the microbiome community. These two samples have been downsampled for simplicity to 10,000 read pairs per sample. And all of this data is available online, and the tutorial to go along with it is available online. We start by activating the Conda environment for QIIME2. Once that environment's active, we can install ITS Express with Conda. ITS Express is the tripping software. The QIIME2 plugin is actually installed by pip, so you need both ITS Express and Q2 ITS Express, the QIIME plugin. In this case, it was already installed. If it wasn't, it would download it. So we can do QIIME dev refresh cache, and what that will do is update the plugins that are available to QIIME. Next, we can check that ITS Express was installed correctly by doing ITS Express help. And you'll see a main menu and the subcommands that are available. So next we can import the data using QIIME tools import. The sample type is paired in sequences with quality. The input format is going to be paired in FASTQ manifest format. That's what that manifest file was for. We use the input path for the manifest file, and then we output it to a QIIME sequences.qza object. After we do that, we can see this file is available. We could summarize that file using QIIME demux summarize command, and then we input the sequences.qza file, and we output the sequences.qzv. QZV is a visualization format, and it allows us to look at the data in the QIIME viewer, which is a really nice web viewer. Yep, the file's there. So we switch over to the QIIME viewer, and we can upload the file. It's actually just kept locally, but we can view the file that we generated. If we click on the second tab, we can see the interactive quality plots. The quality looks pretty good. out to about 220, 230 for both the forward and the reverse read. There's additional summary information in the charts below. The next tab lists some basic information about the file type, and the third tab lists what's the data provenance. So it's all of the processes that have been performed on this data object. That gets pretty useful when you're a little further down the process. So at this point in the analysis, you're going to be trimming the ends off of the ITS regions. And to do this, we'll be using software ITS Express. ITS Express was developed in my group, and it was developed to solve a specific problem. So there was existing software, ITSX, that could trim the ends of these reads. However, as people moved away from doing OTU calling and into exact sequence variant calling, this new method required that every read that was fed into it have all the quality scores, and that wasn't really possible with the old software. So ITS Express solves this. It takes FASTQ files, and what it does is it merges them together using VB-Tools, and then it clusters them at either 100% identity or at 99.5% identity, and it takes those read clusters, runs them through HMM search, finds the end, and then it parses all of the data, and either returns you merged reads or unmerged read pairs that have been trimmed to just the ITS region. So the software is pretty fast. Because of the clustering step, it doesn't have to process nearly as many reads as ITSX did. And so it's about, in the ITS1 region, at 99.5% identity, it's 14 times faster, in the ITS2 region, about 23 times faster. At 100% clustering, it's still faster, 6 to 9 times faster in that case. And the results are fairly comparable because the same HMM models are used as in ITSX. We'll use ITS Express to trim the reads. So we'll use trim pair output unmerged as our subcommand. We'll feed in the sequences.qza file we used previously. And we'll select the region as ITS1. We'll set the taxa to F for fungi. There's 17 other taxa types that can be processed with ITS Express. We'll set the cluster identity to 1.0. That means exact clustering. And we'll set the number of threads to 2. Well, more threads means you can use more threads on your processor. And we'll output the trimmed reads as trimmed exact.qza. So it'll take a few minutes to run, and then we'll write trimmed exact out. The next step in the workflow is calling exact sequence variants. This step takes a few minutes, so I'll tell you a little bit more about it. If you're familiar with OTUs, that's the way that traditionally we've handled error noise that comes through in amplicon sequencing. And so the old way to do that was to simply take the reads, sort them in order of abundance, and cluster them in a greedy fashion at 97% identity typically. And while that works to get rid of noise, it also gets rid of some real biological variation that's in your sample. So a few years ago, several groups came up with statistical methods to handle this, this error problem. And Dada2, which is what we're using, does this by measuring the error rates for all of the transitions between nucleotides. And it builds an error model, and then it is able to statistically estimate at a certain threshold whether or not a new read is an error, is generated by an error, or represents a true real biological sequence. And so that software from Callahan et al. Is pretty effective. There's other software that's built in to QIIME 2 called dblur. I would recommend using QIIME 2 at this point because dblur requires single-ended reads, and it requires them to be uniform length, and ITS region is pretty variable in length, so that has some limitations. At this point, we're ready to move on to calling sequence variants. So we'll use the command QIIME Dada2 denoise paired. For input, we'll use that trimmed read pair sequences that ITS Express generated in the last step. And the next settings determine the trim length of the 3' ends for both the forward and the reverse reads. We're going to set those to 0 because our quality scores are pretty high, but you should always check your quality output plots at the beginning and make sure that, if you have low regions, you trim the ends off of them. That's supposed to help with data 2 clustering or sequence variant calling. And then finally, after we do that, we're going to designate our output directory. And in that directory, we're going to have several different files. We can take a look at these files. A table.qza file is going to have the counts, the representative sequences, has the actual FASTA file, and then denoising stat test stats from the run. Next, we can visualize this data in a little bit more detail using the QIIME viewer. So we'll start a new window, and we'll do QIIME feature table. And we'll summarize. We'll select that output that we just created. Use the table first. We'll select the visualization file that we want to create. It's going to be table.qzv for visualization. And once that file is created, we can go ahead and take a look at it in the QIIME viewer. It takes just a few seconds to generate the file. All right, there it is. So we can take a look at the QZV file in our QIIME viewer. And the first thing we'll see is a per sample summary. So this will be like the number of median OTUs per sample. And then the next area looks at sort of the distribution of counts within your OTUs, or sequence variants. The next plot allows you to look at cutoffs. If you're planning on doing any kind of subsampling, this plot's helpful to sort of see when samples drop out as you subsample. And the third tab looks at individual sequence variants that are called and the frequency of those sequences and the number of samples that they're present in. Now that we have our sequence variants, the next step is to assign taxonomy to those variants. There are a number of ways to do this. Many of you may be familiar with the RDP classifier. It's a naive Bayesian classifier. QIIME 2 uses a similar approach. It uses a naive Bayesian classifier. However, it implements its own version of this. And so the way that this works is you first have to train the classifier. So data is downloaded from the UNITE database, which is really the main database for fungal ITS sequences and species hypotheses. Once the data is downloaded, it takes about 10 minutes to train the classifier. And then once you've got the classifier trained, it only takes a few minutes to actually classify the reads. And what you get back is a listing of the taxonomy broken down at different levels. And that's then put into the OTU table or the sequence variant table and can be used for downstream analysis. And I did want to show you the UNITE database website real quickly and just point out that it does have a download area for files that are formatted for QIIME 2. To use the UNITE data, we first have to import the sequences and the taxonomy information. So we use QIIME tools import feature data taxonomy. For the input format, we use headerless TSV taxonomy format. We use an input path of that file we downloaded and an output path of UNITE taxonomy. It takes a few minutes to load that file. We have to do the same thing for the FASTA file as well. This type will be different. It'll be feature data sequences. The input path will be that other file we downloaded that is of the same type dynamic. And for the output path, we use unite.qza. Next, we are ready to do the classification. So we run the command QIIME feature classifier fit classifier. This is doing the training of the classifier, the naive Bayes classifier. So we load the reference data. That's the unite.qza file we just generated. we feed the classifier the reference taxonomy that we created as well. And then we specify an output for the classifier. When we're done training the classifier, the data will be saved to classifier.qza, and we're ready to start the classification process on our data. We use QIIME feature classifier, classify sklearn. Sklearn is the Python package that the RDP classifier is implemented in by QIIME. We specify the input classifier as that classifier we just trained, and we specify the input reads as the representative sequences from the .2 calling. We only need to classify each representative sequence. The classification result is written out to taxonomy.qza. Next, we can generate a summary of that classification using the command QIIME metadata tabulate. We specify the input file as that taxonomy file we just generated. Then we specify the visualization output, and that file is going to be taxonomy.qzv for visualization. It takes just a moment to process that data and it will be written out. Again, we can use the QIIME taxonomy viewer to view that data that we just generated. So we'll go in and open that file, taxonomy.qzv up, and it's going to have several pieces of information in it. For each sequence variant, there's going to be an ID call, and then there's going to be the taxonomic assignment. You can see some taxonomic assignments are very specific down to the genus or species level, and some are only made at the kingdom level. You can see there's also a confidence score assigned to the assignment. And some of these confidence scores are quite high, others are low, a good bit lower. The data is also exportable in TSV format, so you can click this TSV tab and you can export that data and take a look at it in a text editor or do other things with it, even if you want to do analysis outside of QIIME. We can further summarize our data taxonomically by generating some bar plots. So we use the QIIME command QIIME taxa bar plot. As the input table, we use that table from Dada. And then we add the taxonomy information that we generated. We add the metadata file. In our case, there's not a lot of information in the metadata file, but you could have environmental metadata information such as soil pH or conditions like that could be visualized. And then we generate an output file like taxa bar plots, QZV, the QIIME visualization. And we can look at that in the QIIME viewer. So if we'll switch over to the QIIME viewer, we can upload that or input that file that we just generated and take a look at the results. Initially, we'll just see a bar plot at the kingdom level, which isn't too interesting since it's only fungi we have, but we can select any level we want. So we go to taxonomic level 5. We can see more detail. The sample appears to be dominated by just a few organisms, but there is a wide range of other organisms if we go down a little further in diversity. We can again set that level to a little bit higher if we want a more summarized view of the data. Multiple export formats are available. You can export the data as CSV format, or you can export an SVG format with just the bar plots or with the legend as well. All right, so at this point in our analysis, we've kind of come to the middle point. So we've taken our raw sequence data and we've transformed it into a count table that has sequence variants with the taxonomy called and counts for each sample. This is really kind of the halfway point in the experiment. What you do next depends on the research questions that you have. There are, oftentimes you'll see that people will, at this point, call alpha and beta diversity and kind of look at bar plots. That's sort of standard, but there's a lot of things you can do to kind of go beyond that depending on your research questions. There are statistical tools like ANOSEM and Igneous that allow you to test hypotheses. Igneous is an interesting new tool that uses balance trees to look at the relative compositions of subgroups of taxa across your samples. There's also, if you're interested in the interactions of microbes, there's some nice tools that take into account the compositional nature of the data, like Speakeasy, to construct trees that don't have some of the artifacts that can occur with covariance trees, covariance networks rather. And it is important to remember that this data is compositional count data, and so there are some things you can't answer with that kind of data. The total number of counts in a sample doesn't matter because it's compositional. And it's important to also remember that simply dividing by the total doesn't solve the problem of compositional count data. You need to remember that there are certain things that can't be answered. So compositional count data, each sequence, the count of each sequence variance depends on the count of all of the other samples, all of the other sequence variants in the sample. And so the count of a specific sequence variant in sample 1 can be higher than in sample 2, but it doesn't necessarily mean that there's more in sample 1. There are some transformations that can be done to analyze this data, but it's important to just remember that you're working in not a real Euclidean space. You're working in a compositional count data space. So hopefully that gets you started on your fungal amplicon sequencing workflows, and that you'll be able to kind of use this as a jumping off point to answer the scientific questions that you have.